This session of the 6th International Chromosome Conference, also called the Helsinki Chromosome Conference, has been entitled Past and Future of Chromosome Research. Today is the 30th of August, 1977, and I believe it is only fair to say that so much has been achieved in the various fields of chromosome research that this is an appropriate time to look back and try to find out something about how it all happened. On the other hand, scientists tend to like to look ahead and indulge in speculations about what will happen next. Therefore, the future of our field will also be covered. Our first speaker is Professor Darlington of Oxford, who obviously needs no introduction. However, I should like to remind you that he is not only the grand old man of chromosome research, but also the founding father of these international chromosome conferences, which started in Oxford in 1964 as the first Oxford Chromosome Conference. The title of Professor Darlington's introduction is The Chromosome Revolution.
Mr. Chairman, thank you for your kind words. And thank you also for giving me a chance of speaking on this occasion to this conference and on this subject. I have something to say which I should not and would not like to say anywhere else, and that is why I am particularly grateful. At the beginning of this year, a book was published in America by Ian Shine, a resident of Lexington, Kentucky. The book was a life of Thomas Hunt Morgan, and I will write down his name, and that is the date 1977, because many will probably like to look at this book later. There is much in this book that I did not know, and there's much that I did know that wasn't in this book. The consequence was that a very rapid flow of ideas went through my mind, the results of which I am going to try to give you. I had to go back and look at the whole history and meaning of genetics and of the chromosomes in order to reconstruct my own ideas and tell you what the result was. Now, our subject is now 100 years old. It began in 1875, a year in which the meaning of fertilization was discovered to lie in the fusion of nuclei, and the meaning of nuclear division was found to lie in the division of certain longitudinal and exact division of certain threads. That was really the beginning of the thing. But the great events in the history of our subject occurred between 1882, which was the year in which Darwin died, and 1910, when Drosophila was discovered. It was during this period of 30 years that the chromosome revolution of which I am going to speak actually took place. But the great year the great year and a half in this period was the 1887 to 1888. And in this period of 18 months, two great milestones of our of our study were laid down. The first was when the professor of zoology in Berlin, Waldeyer, gave a review of what he believed to be the relevant problems of cell division. He said, we need a word to cover these split threads that Fleming has talked about, and that these chromatic elements that Boveri has described. We need one word to describe both these things, because they are one thing, and I propose that we should call them chromosomes. That, of course, was an easily forgotten little triviality, as it might appear, in the development of the subject, but it was not a triviality at all. For in this review, Waldeyer, which it's extended over 120 pages of print, in this review, Waldeyer gave us, and gave the world, an argument, a generalization, and a theory, the chromosome theory. He showed that the chromosomes were responsible for the activities of the cell, for the development of the organism, and for the heredity of the race. That was quite a lot. And it wasn't missed. The meaning and importance of it did not escape the wide Western world. Immediately it was translated into English, and it appeared the following year in the leading English journal on microscopic science. This article, this statement of the name of chromosomes, was a manifesto to the whole world of naturalists that something new had appeared. And it was not only a manifesto to the world outside, it was a program for the people who were working on these chromosomes. Now, you must realize that all this business that I've been talking about came from Germany. It came from German microscopes and German chemistry. German chemistry provided the dyes, German chemistry provided the idea of nucleic acids, and the German microscopes, of course, provided the means of looking at these things. The whole business had to spread out from Germany, which it did, encountering resistance at every stage of its progress. Then 5 years later, in 1892, and it's very important to remember these dates because it makes all the difference in the world whether one man says a thing before or after another man. In that year, the professor of zoology, I think, at Munich, whose name was Rickert. Now, I like to do honor to him because hardly anybody else has done. Rickert looked at the chromosomes, the lamp brush chromosomes, in the developing egg of a shark. And he saw certain figures with which we are now today very familiar. They look like this. And he said, these chromosomes are meeting at certain points. Those points he called points of Überkreuzung. Now I wonder where we've seen that word before. Überkreuzung. Crossing over, I believe. And he said, now, going back to what Weissman and Boveri had said, those points of Überkreuzung here must be points at which material is exchanged between the chromosomes. You see, he was perfectly plain about his meaning. He said there must be a Substanzaustausch. an exchange of substance. And you see, he got an idea there. It's still very much with us. That was in 1892. And during the following years, people, of course, sorted out meiosis and knew just what these things were doing. They knew that, in fact, the chromosomes at these stages were double. Something like that. And they knew how they paired first side by side, and they knew how afterwards they segregated, and they followed. During the next 17 years, all these things were sorted out, and names were given to all the stages and everything else that we know about. Now, Rickert said, now this idea of a Substanzaustausch is an Ergänzung of a corollary, a completion of Boveri's idea of the continuity of the chromosomes, because you must have an exchange of material. If, for example, in Ascaris megalocephala you have only one chromosome, you don't have any exchange, and you don't have any recombination unless there are places of that kind. So this was necessary in order to put together all the ideas of the great pioneers of our subject in Germany. Now, these things went on, and in due course, 17 years later, the Belgian cytologist Janssens came along in 1909. Janssens. And what did he do? He referred to Weissmann. He referred now also to Mendel, because Mendel had been rediscovered. And he also said that these there must have been exchanges of material. But unfortunately, Janssens didn't mention Rickert. But his paper was read by Wilson. And Wilson in Columbia University in New York took the paper through. I'm pretty sure, I've been in the rooms myself, took the paper through to Morgan and said, "What do you think of this? This seems to explain what you're doing." And Morgan, of course, after a little thought, decided that it did explain what he was doing. Now, you see, the chromosomes do what genetics requires them to. Well, not genetics exactly. No, the chromosomes do what the organisms require them to do. Now, this is a very interesting idea. Just look at it. Here are the organisms. And here are the genes. This is the theory of the gene. It's a very important theory because it was taken up by a lot of other people. Now, the organisms were connected with the genes, and the genes were connected with the organisms. And in between, there were some shadowy things called chromosomes. Now, I'm not exaggerating, because those chromosomes were non-experimental entities which did what organisms and genes required. They were abstract. No, they didn't. Nobody wanted to look at them if they could avoid it. And they didn't. Actually, meiosis, of course, was fortunately in Drosophila was quite impossible, and nobody bothered to look at meiosis in other organisms to see what that was doing. That was being done elsewhere by the chromosome people, and there was very little connection between the two. So I would say that the Morgan model that had been produced represented an organismal point of view. The point of view of the naturalist the point of view of Darwin, which was almost sacred by this time, the point of view of the Darwinians, the point of view now of a new generation who came along, who came to be called the Neo-Darwinians. Now they, of course, had not seen the chromosomes at all. It's wrong to say one or two of them had seen the chromosomes down somebody else's microscope, but for a very short view. Now, let me take the leading Neo-Darwinian. How did Fisher describe the chromosomes? Well, he followed very closely the pattern that Morgan had provided for him. The words he used were, and this is 1930. Let us be quite certain of the date. We're now on to 1930. Fisher. He said, there are dark staining bodies or chromosomes, which are to be seen in the nuclei of cells at certain stages of cell division. And there Fisher was prepared to leave them. And I must say that Haldane was, although working so closely with me, he couldn't speak quite in such casual terms. Nevertheless, from his point of view, it was very much the same. They believed, in fact, that the chromosomes were doing what genetics required. And the chromosomes were not genetics. They were, I think the word now used, which I much object to, is cytogenetics, a sort of second-class genetics, which isn't fundamental. It's not fundamental, it merely rests on things you see through the microscope, if you have a good enough microscope and a lot of patience. Now, we know, and Wilson knew 70 years ago, that the chromosomes, in fact, show all kinds of evolutionary properties which are very different from anything seen by organisms or recognized immediately in experimental processes. We know that the chromosomes show evolutionary trends. That the sex chromosomes, for example, go through long-term changes, which you can recognize and which Wilson illustrated in 1907, or they develop complex heterozygotes or